Aloha and welcome to our video on transcription and translation. The goals for the video are to describe transcription and RNA splicing. We're also going to describe what translation is, and we'll give you a couple examples of mutations. Okay, let us begin by talking about transcription. Transcription is the process of transcribing or changing our DNA, which is the blueprint, into something usable. And that usable thing is going to be RNA. And in fact, we're going to make messenger RNA, and that messenger RNA is going to be able to travel outside of the nucleus into the cytoplasm, where it'll meet up with some ribosomes, and that's when we can make the actual proteins. So this process is going to go how we get from DNA to RNA. Okay, so to begin our process, we have to start with the DNA. Remember, the DNA is in the nucleus. It's a double-stranded molecule, and this is our genetic material. This is what is the blueprint to how we build our cells. So this has the codes for all of the different proteins that we'll need to make our cells. And we need to change this DNA to RNA so that we can take it out of the nucleus and start manufacturing those proteins. Much like replication, you'll see that we're forming a little bit of a bubble here. However, we are going to do it a little bit differently. Here we have a promoter, which is going to separate the two strands of DNA, and then we have a terminator, which will end this separation. So instead of separating it and making a whole complete strand, we don't need that. When we're talking about making the RNA to make a protein, we're going to have a section of the DNA, a chromosomal gene per se, and that section of DNA is what we're going to copy off of. We don't need the entire thing. Okay, much like the replication, in transcription, we'll see that inside this bubble, we're going to have this molecule, RNA polymerase, is going to go along the DNA molecule. So this is our DNA molecule, and it's going to serve as a template to make the RNA. So now as we're going through, we'll match up our base pairs. Okay, here we have a guanine matches to a cytosine, and a cytosine to a guanine, and a thymine to an adenine. But notice here we have our adenine, and now it matches up with uracil instead of thymine. So this uracil is what makes it that RNA. Okay, and we're forming a single strand of mRNA. And remember, RNA is going to be a single strand, DNA is a double strand. After we've created the mRNA, the DNA will close back up and it'll go back to doing what it's normally doing, probably going to open up again and make more messenger RNA. Might be the same gene, might be a different gene, but that's how the process goes. And we're left with this mRNA molecule. And remember, the mRNA, unlike DNA, is a single strand, and it also has this uracil instead of the thymine that we saw earlier in DNA. Okay, so now that we have the messenger RNA, which is the template for making a protein, we have to make the protein. And that process is called translation. Now, translation happens in the ribosomes, and the ribosomes are made up of ribosomal RNA and some proteins. And we'll see what these do in a little bit. We also have this transfer RNA molecule, and the transfer RNA molecule has an amino acid attached to it. And when we string these amino acids together, we form a protein. Now, the mRNA is going to have its bases broken down into these groups of 3 that they call a codon. If you notice with the transfer RNA, we also have a group of 3 here. So this codon tells us which amino acid we need to make the protein, and the transfer RNA will bring the proper amino acid. And the way that it brings the proper one is only its anticodon, or its 3 base pairs, will line up and make a match. Okay, so let's go through the steps of translation. We're going to start off in the nucleus. Remember, inside the nucleus is where we can find our DNA. Now, our DNA has gone through the process of transcription, and it has made this messenger RNA, and this messenger RNA is able to exit the nucleus through these pores. And that's what gives us the material out into the cytoplasm so that we can start building proteins. Now, all of our proteins will start with a start codon. And remember, the codon is our 3 bases. Okay, it's a little system like that. So those 3 bases will come together. The start codon for all of the proteins is going to be this one right here, this adenine, uracil, guanine. Okay, so that will come and that will line up into the ribosome, and the ribosome will take the mRNA and feed it through in this direction. So it first starts off with this start codon and it says, hey, we're gonna start making proteins. And that's what the start codon does. Now, if you remember, we have our start codon, which was these 3 base pairs here. We then bring in our tRNA molecule. Now, our tRNA molecule has the anticodon, which is going to be the 3 complementary base pairs for the codon. And you'll see that the adenine and uracil bond up, and the cytosine and guanine bond up. And this one here, because it only matches up this way, it brings in one specific amino acid. And that amino acid begins to be the first of many in our protein polymer. Okay, so here we see the end of the process. So we missed this middle part. Remember, the mRNA has traveled through the ribosome this way, and we have this broken down into our 3 little codon systems here. Okay, and what these 3 little codons do is, remember, they line up with an anticodon on a tRNA molecule, and they bring in a specific protein. So we can see how these specific proteins are all lining up together to form this protein polymer. When it gets to the stop codon, and there's several different stop codons, and this one, it's this UGA, what that's going to do is that will tell the ribosome to stop bringing in more. That's the end of it. So even if this chain went a little bit longer, what it's going to do is here, when we attach this one, it cleaves it off, and that ends our protein for us. So now we have our completed polypeptide, our protein. The amino acid chain has been released, and it becomes this polypeptide. And then, because of the hydrogen bonds between these different amino acids, it'll start to fold and take shape and become the protein structure that's necessary for the cell to do what it needs to be doing. Now, sometimes mistakes happen and we have a change in the DNA sequence. And that can happen in the DNA, or that can happen during DNA replication, where we can make a mistake, or it can happen in the transcription when we're making the mRNA molecule. And what we notice is we have our DNA here, and it kind of shows us what it is. Now my DNA is going to match up to my mRNA, which then matches up to my tRNA, and then we have our amino acids here. So we can see how this process is going to make this protein that'll look this way. Now, the easiest type of mutation, or the one that we see quite often, is this substitution, and that's where you just make a mistake. So here in our example, if we look at it, we have our first codon is still the same, our second codon is still the same, but in the third codon, what happened was we took this C out and we replaced it with a T. So the cytosine went out and the thymine came in, so we have a little bit of a difference here. Now, because it's different, it's going to bond to something different here, this A, and we notice that here in the original protein, we had a CGU, which formed arginine, and here we formed histine. Okay, so we can see that we have this little bit of a difference, and that's going to form a difference in the protein. Sometimes it's noticeable, sometimes it's not, so it's going to vary, but our first one is where we're just substituting one for another. Now, there's 2 other kinds of mutations that happen every once in a while. One is a deletion, where we'll actually just be going along and we'll actually just forget this one. Okay, so it's just deleted out of there. And then its opposite is going to be this insertion, where we'll just add in an extra one also. And these are our major types of mutations that we see. And you can see how they're all just going to be mistakes. So even in the conservative, it's semi-conservative replication and semi-conservative transcription, it's because we're using one as a template. Every once in a while, we'll still make mistakes. Okay, so the lessons will go over these in a little more detail. As always, good luck with those, and we'll see you in the next video.